Hello, BISC1323. This is the beginning of recorded lecture 1-2. Moving on to viruses. So here's a phylogenetic tree that we looked at last time. This is the real zoomed out version showing all life on the planet. Bacteria, archaea, eukarya were our three domains. And if we look closely at this, we can not see viruses anywhere. Viruses are not on this phylogenetic tree of life because, importantly, viruses are not living things. You may remember this slide from BISC1323 going through the properties of living things, and viruses do some of these, but they're not made of cells. We don't consider viruses to be alive, so they don't have a place at all on this phylogenetic tree of life. So what are they? Well, they're non-cellular, they're not alive. We can call them parasitic macromolecules. It's kind of a mouthful, but it's a good way of describing these. And importantly, they are much smaller than cells. Here's a very interesting scale here. Human, ostrich egg, chicken egg, more eggs here. But yeah, now we get to eukaryotic cells around this size, bacteria about the tenth of the size of eukaryotic cells. And here's a flu virus. That's one of the bigger viruses, but even that is much smaller than even a bacterium or an organelle. In fact, viruses are so small they cannot be seen with standard light microscopes. So very difficult to detect because they're so small, and yet definitely not made of cells, not considered to be alive. We don't know where these things came from. Their origin is not understood. We know that they're around. We didn't know where they came from. What they are physically is genetic material inside of a protein shell. That shell is called a capsid. It's just the term for the protein case for their genetic material. Importantly, I did say genetic material instead of DNA because while all living things use DNA as their genetic material, there are some viruses that use RNA as their genome instead of a short-term copy. So yeah, some viruses use DNA, some use RNA. But either way, they're genetic material inside of a protein capsid. Let's look at these protein capsids. So there are three common capsid morphologies, shapes that this shell can take. One of these is called a helical capsid. It looks like a tube, but really it's a capsid protein that's repeated over and over and over again, sort of in a spiral here. That's why it's called helical, like a spiral. But it ends up looking like a tube with the genetic material, DNA in this case, on the inside. So helical just means tube. Another common one is icosahedral. This is supposed to be a polygon with 20 sides. It's rarely drawn super accurately, but some sort of complex polygon with genetic material inside. So yeah, this capsid is called icosahedral, some complex polygon. And the coolest looking one is what's called binal capsid, or it's sometimes called head and tail. Looks like a creepy spider or a lunar launcher or something like that. But really, this is a helical capsid and an icosahedral capsid put together. So binal, also known as head and tail, icosahedral head, and helical sheath. There are some other capsids, but these are the big three we got to know. Every virus has a capsid. Some viruses, in addition to this capsid, have a structure called an envelope. Envelope is defined in key terms as a lipid bilayer that encircles some viruses. So yeah, this is again, in addition to the capsid, you've got your genome here surrounded by the capsid, and then there's this envelope that surrounds the capsid itself. This is a pretty big, complicated structure with, again, it's not just a protein repeated over and over again. This envelope has lipids, tons of lipids, and proteins embedded within those lipids. This seems really complicated for a virus to make. Well, spoilers, the virus did not make this, it stole it from its previous host. And again, not all viruses do this, but some of them do have this structure called an envelope. Now, we talked in the previous lecture, in the previous chapter, about trying to put together evolutionary relationships. And yeah, it's true that viruses do not appear on this phylogenetic tree of life, but could we instead try to construct some sort of phylogenetic tree of virusdom or something like that? Well, you can't really do that, because for viruses, what we would want to do is a sequence alignment like this. We want to compare genes, see how closely related they are to one another, or how different they are to one another, to compare flu and polio and smallpox. And if we want to make some sort of tree, we got to line everything up and compare them. Well, we can't do that because they don't have anything in common with one another. We could take for granted in this particular alignment that all vertebrates have this histone H1 protein, so we can all compare them because everyone has this. But when it comes to viruses, they don't have genes in common with one another to do this sort of comparison. Let me give you just a sense of what the genome of a typical virus looks like. So a typical eukaryotic genome, again, we're big, we're complicated. The human genome has about 20,000 genes. Bacteria and microbes like archaea have about 1,000 genes. So definitely smaller, definitely simple. Most viruses have about 12 total genes. Influenza has 8. So these are lean and mean, and they have a very minimum number of genes that they need to do their job. Because they have so few total genes, none of these have something in common that all viruses have, so you can't really make this sort of virus tree. You could try to. Virologists definitely try, but they are very difficult to classify evolutionarily because they don't have stuff in common. That being said, like I said, viruses do try to, virologists do try to construct these sorts of trees. We can try to classify viruses based on their genome structure, whether they use RNA or DNA, their capsid morphology, which one of those three they have, or how they make their mRNA. This is a weird one, but it's probably the most accurate. There are different ways that these viruses go about making messenger RNA, and lumping them together into different groups based on how they do this is probably the best supported way to try to classify them evolutionarily in the absence of being able to do any sort of sequence alignment stuff. So that's enough about virus structure and evolution. What do they actually do? What does a viral infection look like? Well, there are a couple of different ways they can do this, but either way, we classify them as obligate intracellular parasites. We know what parasites are. Intracellular means inside of a cell. So in order to be a parasite, in order to steal resources from their host, they do that by getting into the host's cell. Obligate comes from the word obligation, which means something you have to do. So they have to be intracellular parasites. They have no other way of doing things, of making more of themselves. They have to be parasites, and they have to be parasites that get into a cell. So they require a host cell for replication. They can't do anything on their own. They need a host cell to make more of themselves. So there are two types of viral infection cycle. Let's start by looking at something called the lytic cycle. So let's take this one step at a time. Here we've got this binyl or head and tail capsid here with some genetic material inside. Step number one is attachment. The capsid has to bind to the host cell. And it looks like it would just stick on, really easy, but there are actually some very complicated protein-protein interactions that are occurring between the proteins of this viral capsid and proteins on the surface of this host. This gives specificity to these viruses to ensure that if this is a virus that's meant to infect some species of bacterium, this virus is not going to infect you or a plant or your cat. It's only going to infect this species of bacteria because they have the right proteins on their surface for this attachment to happen. So easy first step, but it's important to note. Step 1 attachment involves specific protein-protein interactions between virus and host. Next, we have entry. There are a couple of different ways this can happen. This says penetration because it's injecting its genetic material into the cell, leaving behind sort of a empty, hollow, non-functional capsid. But injecting the genetic material in is just one way that this can happen. So entry involves injection of genetic material, or sometimes the host cell can take up the entire capsid. Here's a viral capsid interacting with the host cell, and it sort of tricks or forces the host cell to take up the whole thing into a vesicle, and then it escapes this vesicle and wreaks havoc inside of the host cell. That's another way that a virus can gain entry. Or if we're talking about an enveloped virus, enveloped viruses can get into their host simply by fusing that envelope with the host membrane. So here's what this can look like. There's the genetic material, there's the capsid, there's the envelope, a bunch of proteins on the envelope, there's the host membrane. Lipids fuse together pretty easily and form just a slightly larger bilayer, and that releases the capsid and any other goodies that would be inside of this envelope into the host cell for the virus to do what it needs to do. So any of these three mechanisms can get the virus into the cell. Next, replication. The virus is going to force the host cell to make more of its genomes and its proteins. Again, the virus doesn't have the means of acquiring energy on its own. It steals energy from the host. The virus does not have enough genes of its own to do DNA synthesis and transcription and translation and stuff like that. It's stealing those enzymes. It's hijacking that machinery from the host cell to make more of itself. So step 3, replication. Host energy and host enzymes are used to make a bunch of viral proteins and a bunch of viral genomes. Next, we have assembly. So we're going to start building more capsids and putting viral genomes inside of those capsids. This one's easy enough. Capsids assemble with genomes inside. And finally, we have what I'm going to call egress, where the viral capsids are released from the cell. And there are a couple of different ways this can happen. Here we see the cell bursting open, releasing a bunch of these viruses to sort of float around, and hopefully, or they hope, they'll infect some other host cell and keep this going. But another way that we can get release is by budding. Don't sweat all the details here. It's from an immunology textbook, but this is how influenza virus gets its envelope. It has its capsid with its genome inside. It fuses with a segment of the host lipid bilayer and sort of buds off and pinches off part of that membrane. That's how it gets its envelope. So this does not kill the cell. This does not burst the cell open, but this definitely does. So egress is by lysis or by budding. Lysis is defined in the key terms as the bursting of a cell, easy and straightforward, and budding is defined as a method of exit from the cell where viruses leave the cell individually by capturing a piece of the host plasma membrane. Either way, virus is going to get out and try to infect more cells. This has been the lytic cycle, which is sort of the simpler of the two. Now, let's talk about the other one, the lysogenic cycle. So the lysogenic cycle looks pretty similar to the lytic cycle. And in fact, it has a lot of the same steps. You could think of this as kind of an extension of the steps of the lytic cycle. So it begins the same way. We have attachment, and I'm not writing it again, but it's still true that there are specific protein-protein interactions. You have attachment, You have entry, and again, I'm not putting it in here again, but all the different ways of getting in are different ways of getting in. But instead of going straight to making more virus, in the lysogenic cycle, we have a step called integration, where the viral genome will integrate or insert itself into the host chromosome. At this point, viral gene expression is turned off. It's gone quiet. It's not trying to raise the alarm of the immune system or whatever it is. It's not trying to make more of itself. It's hiding. But because it's part of the host genome, every time that genome is replicated, it's going to get replicated as well. So at this point, when the viral genome is sort of hiding out like this, integrated, it is called a provirus, and it propagates as the host propagates. So now you've got two proviruses, two daughter cells that both have this hiding, quiet viral genome integrated into this host chromosome. So integration here comes after entry. Viral genome integrates into host chromosome. Viral gene expression shut off. It's now called a provirus. And propagation, the viral DNA replicates with the rest of the host cell. Now, this can go on for many, many generations, especially if we're talking about free-living bacteria that replicate every 30 minutes. You can get a lot of proviruses as it goes through propagation. The party is over as soon as the host cell experiences any sort of metabolic stress. Maybe the temperature is getting too high, or maybe it's running low on sugars. As soon as the host cell starts to get stressed out, like rats leaving a sinking ship, this virus is going to start expressing its genes again, force the cell to use what resources it has to make more viruses so it can assemble and then get out of this sinking ship. So induction is where stress triggers the lytic cycle steps and viral gene expression is turned on. And then the same stuff we talked about before: replication, assembly, then egress. So yeah, this is the lysogenic cycle, again, sharing a lot of steps with the lytic cycle, but yeah. To give you a couple examples, lytic cycle, influenza. So yeah, the flu is something that affects humans, and yeah, this is not meant to show flu here, but flu works like this. It gets into our cells, it steals resources, it makes more of itself, it gets out. It doesn't stay in your genome, it doesn't get into your genome. It does its damage and then it gets out and tries to get you to spread it. If you want an example of a lysogenic human virus, the herpes simplex 1 or 2, so genital herpes or cold sores. I don't have a slide for that. You can Google medical pictures if you want to on your own time. But cold sores are a recurring thing because that herpes simplex virus integrates into the genome of these neurons in your mouth. And then, yeah, they're called cold sores because when the weather turns cold, or you're stressed out because it's finals week, or your cells are experiencing some sort of stress, the virus is going to go back to lytic cycle and a fever blister or cold sore will pop up. So those are just a couple of human examples. An important thing to note when it comes to viruses is everything has a virus. All living things on the planet, animals, plants, bacteria, everything out there has to deal with viruses. There are viruses that infect us, obviously. There are viruses that infect any animal you could think of, vertebrates, invertebrates, viruses that infect plants, viruses that infect bacteria. Everything has viral parasites. However, each virus is specific to a certain range of hosts. I mentioned this in the first step of attachment, that this ensures that a virus that infects a plant is not going to infect you because you got different proteins on your surface. So yeah, each virus is specific for a certain range of hosts. Some are a little broad, most of them are really specific. A good example of this is something called bacteriophage, also known as phage. This is just a name for a virus that infects, any guesses? Bacteria. Yeah, the key term says a bacteriophage is a virus that infects bacteria. Just an example of one with a specific host range. Now, the textbook has a big section on plant viral infections, and yeah, importantly, these are all plants that we care about, whether it's lettuce or peanuts or sugar cane or cauliflower. I'm not going to make us memorize all these. I just want to point out many viruses are responsible for plant diseases, and they cause pretty significant economic impact. There's also a big section in the textbook about human infections. And once again, I'm not going to make you memorize a laundry list of different human viral infections, although it's, certainly interesting to stare at this for a few minutes, I guess, and see how many of these you've had in your lifetime. What I can say is that many viruses infect humans, and yet together these have a very significant impact on public health. What I do want to say about these in general, I want to say a little bit more than just this, is most of these viral infections do not have a cure, like a medicine, a pharmaceutical that you take that makes this infection go away. Most of these, you treat the symptoms of the viral infection, and it sort of runs its course. So most viruses, there's rhinovirus. When you take medicine for the common cold, it's just treating the symptoms. You're waiting for it to just burn itself out. Same thing with a lot of these gastro, viruses. You're just treating the symptoms, staying hydrated and stuff like that, just letting the virus burn itself out and do its thing. So antiviral compounds are not widely available for most viruses. This is because most of these viral infections are self-limiting, which means they get in, they do their damage. That doesn't mean they're not deadly. Many of them are, but they're gonna, limit their infection and cause the damage they're gonna cause, and then get out. And so, instead of having antiviral compounds, a lot of these things that you take for a viral infection treat the symptoms. A more promising avenue for trying to limit the public health impact of all these horrible viral infections is not necessarily antiviral compounds, but vaccines, which have been very effective in preventing infections in the first place. We'll talk more about vaccines later in the quarter. We've got a big chapter on the immune system. You got to understand how the immune system works before you can understand how vaccines work. But measles, mumps, influenza, polio, human papillomavirus, rubella, smallpox, a couple of different types of hepatitis. There are many viral infections that have vaccines that effectively prevent them. So that's what I wanted to say about viruses and human health. Now, for the next section, I want to present this a little bit differently, sort of like a mystery. I want to talk about 3 neurological diseases. The first one up is bovine spongiform encephalopathy. Nailed it, first try. Also known as BSE, also known as mad cow disease, because those are all easier than saying bovine spongiform encephalopathy. Mad cow disease causes a lot of neurological symptoms in cows, progressive degeneration of nerve tissue, including the brain, ultimately leading to death. Really bad. There is a similar disease that affects sheep called scrapie. Part of these neurological symptoms involves a compulsion to scrape themselves against fence posts and barns and stuff like that, hence the name scrapie, but it's also just progressive neurological breakdown. The third one is one we find in humans, called Kuru, also known as Creutzfeldt-Jakob disease. To show you what I mean by progressive neurological breakdown, normal brain, Creutzfeldt-Jakob brain. Just every single part of this looks shrunken and deteriorated. The symptoms are, widespread and varied because everything is just being destroyed. So you might assume that these are caused by a virus because we're in the virus chapter, right? Well, let's pretend like this is an episode of House or something like that, and then if you have some sort of patient with these symptoms, what can you try to rule out? Well, we can rule out toxins and poisons, because lead poisoning does, a lot of these similar symptoms, but we know it's not a toxin, we know it's not a poison because it's infectious. Kuru or scrapie or mad cow can be transmitted from infected individuals from their nerve tissue. And this is why, BSE in particular is so important for us, because these are food sources. And yeah, we're not eating too many cow brains, but there is nerve tissue in muscle tissue. They're connected together. So yeah, eating meat from an infected cow will pass this on to a human. So yeah, this is not a toxin or a poison because it can be transmitted. It's also not a virus. So yeah, I'm throwing you a curveball. We're in the viruses chapter, but we're not talking about a virus. We know these diseases are not caused by a virus or even by a living organism. So it's not a bacterium, it's not an archaea, it's not a eukaryotic parasite. We know it's not any of these because we can take that nerve tissue, irradiate it. This is a process that destroys DNA and RNA, so it doesn't matter if you're an RNA virus, destroys DNA and RNA, and that nerve tissue still transmits disease. So this was a real puzzle for a long time about what was responsible for these diseases. What we now understand is that Kuru and scrapie and mad cow are caused by a protein, specifically a misfolded protein that we call a prion. It's thought to behave like this. We all have, or all vertebrates have, prion protein, normal prion protein present in our nerve tissue. However, the disease version, it's the same protein, it has the same amino acid sequence, it's just folded in a different way. And this misfolded version comes into contact with the normal version and converts that normal prion protein into more misfolded prion protein. As more and more of the normal stuff becomes abnormal prion protein, that's what's causing the progressive, just across the board neurological breakdown. This also explains why it is infectious. If you eat meat or something like that has this abnormal version, well, it can start converting your normal versions into more abnormal versions. So the terminology here is a little dicey, but bear with me on this. So we all have prion protein. We call the normal version P, capital P, lowercase r, capital P, superscript C. The C stands for control, but it's the normal version that we all have. The disease-causing protein is capital P, lowercase r, capital P, superscript SC. It's named after scrapie. Again, the names are so similar because it's the same protein. It's just folded differently. When PRPSC comes into contact with PRPC, it converts it to PRPSC. And lots of this PRPSC is responsible for lesions in the brain. And again, this is not a virus. It's in the virus chapter because it's this sort of other acellular entity. So like a virus, these prions are acellular. They're not living things, but they are responsible for some pretty significant and important diseases. There's actually one other acellular entity in this chapter. I won't present this one as a mystery, but another acellular entity is something called a viroid. A viroid is like a virus, but it doesn't have a capsid and it doesn't have an envelope. And you should remember that a virus is just genetic material in a capsid and maybe an envelope. If you're talking about something that doesn't have an envelope and it doesn't have a capsid, isn't that just genetic material? Well, Yes. Here's a sequence of RNA that is the potato spindle tuber viroid. Again, it's not much to look at because it's just a sequence of C, G, G, A, C, U. It's just a sequence of RNA. But despite not having a capsid or an envelope, it's still able to cause disease. These are potatoes that are all messed up with potato spindle tuber viroid. It's still able to do all the things that a virus does. So it's just RNA, and they replicate in the host cell just like a virus does. And the only viroids that we know of infect plants. There are no known viroids that infect humans or any animal at all. But potato spindle tuber viroid is probably the most famous of these. But yeah, you should know what a viroid is, and you should know that it only infects plants. Okay. That's the end of this virus chapter, including these sort of bonus other acellular entities. And that also brings us to the end of recording.